Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on the factors that affect enzyme activity. Last time, we discussed the effect of enzyme concentration. We learned that the rate of an enzyme-controlled reaction is directly proportional to the number of enzyme molecules. We also discussed that an enzyme's activity is limited by the concentration of the substrate. When there is a limited supply of substrate, an increase in enzyme concentration has no effect on the amount of product formed. In today's lesson, we will look at the effect of temperature on enzyme activity. If you are ready, students, let us begin. In the previous lesson, we studied the production of biofuel. We learned that cellulose is most easily broken down into biofuel when it is heated to temperatures of 70 to 80 degrees Celsius. There was a problem with carrying out this reaction at such a high temperature. Do you recall the problem, students? Please write down your response.
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Hello, students. Although cellulose is more easily broken down when it is heated, cellulase, the enzyme that breaks down cellulose fibers, can only function up to temperatures of 50 degrees Celsius. Well done, students. When the temperature of a reaction is raised, particles of the reactant are given more kinetic energy. This means that free particles move around more quickly, thus increasing the probability that a substrate particle will collide with an enzyme molecule. More collisions lead to the formation of more enzyme substrate complexes and, therefore, a higher turnover rate. An increase in kinetic energy also means that the particles within a molecule will vibrate more energetically. These vibrations put strain on the bonds that hold the atoms in place. Bonds begin to break, and, in the case of the enzyme, the shape of the molecule begins to change. This change causes the active site in particular to change shape. In a previous unit, you learned that all globular proteins have a unique tertiary structure, which gives them a unique shape. When this structure is altered, we say that the enzyme has been denatured. In a laboratory, biologists often conduct experiments to determine the optimum temperature of an enzyme-controlled reaction. Students, how would you define optimum temperature? Please write down your ideas.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello, everyone. Please compare your definition to this one. Optimum temperature refers to the temperature at which the number of collisions between enzyme and substrate particles is greatly increased, and the enzymes have not been significantly denatured. If the raised temperature results in little denaturation, but a greatly increased number of collisions, the activity of the enzyme will increase. If the higher temperature causes significant denaturation, then, despite the extra collisions, the activity of the enzyme will decrease. Optimum temperature is the balance of these two effects, in which one does not outweigh the other. As you can see in this graph, students, the effect of temperature on enzyme activity is not symmetrical. As the temperature is raised, there is a gradual increase in enzyme activity. The peak of the graph represents the optimum temperature. Above the optimum temperature, denaturation occurs very quickly. As we can see, the graph drops off very quickly. Once the active site of the enzyme has completely changed and no enzyme-substrate complex can be formed, the reaction rate is 0. Every enzyme has a different range of optimum temperature. For instance, students, the enzymes in some plants growing in the Arctic may have an optimum temperature that is less than 5 degrees Celsius. In comparison, some thermophilic bacteria that live in hot springs have an optimum temperature over 90 degrees Celsius. There are hundreds of enzymes within the human body. Most of these enzymes have an optimum temperature of 37 degrees Celsius. Students, do you know the significance of this temperature? Let us pause for a moment while you discuss this with a classmate.
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Welcome back, students. Were you able to determine the significance of 37 degrees Celsius? This temperature is the same as normal body temperature. Also, this is the temperature at which human body enzymes function best. If you answered the question correctly, well done. Most enzymes in the human body will shut down at a body temperature below or above a certain value, although this value can vary slightly according to each individual. This happens during hypothermia, when the body temperature drops too low, and during hyperthermia, when the body temperature gets too high. Fortunately for us, as long as the enzymes are not exposed to higher temperatures for too long, denaturation is usually reversible in living cells. Let us use the example of boiling an egg. As an egg is boiled, the liquid albumin protein solidifies and turns white. The enzyme structure has been permanently broken, and the protein has been completely denatured. You cannot unboil an egg by cooling it. This temperature effect is not reversible. Students, in today's lesson, we have discussed the effect of temperature on enzyme activity. We have learned that when a reaction is at its optimum temperature, the increased number of collisions between substrate and enzyme particles balances the effects of denaturation. We also discussed the range in optimum temperatures that occurs in different enzymes. We learned that in living cells, denaturation can often be reversed, but the same is not true for non-living cells. Next time, we will learn about the effect of pH on enzyme activity. Until our next lesson, thank you, teacher. Thank you, students.